Good afternoon. It is my pleasure to introduce Tina Stankovic, a woman who is a neurotologist and auditory neuroscientist who needs absolutely no introduction to this audience, so I will make this very brief. After education and PhD in Boston at Harvard and MIT respectively, she did her clinical training at the Mass Eye and Ear. In 2022, moved across the country to become the chair of the Department of Otolaryngology, Head and Neck Surgery at Stanford. Notably, recently she was inducted into the National Academy of Medicine and was also on the Huberman podcast. By the way, an excellent episode if anyone hasn't seen it. So she's going to speak to us about the inner ear. Thanks, Tina.
Thank you for the kind introduction and for the opportunity to tell you about our work. I would like to disclose the funding for this work from the government, several foundations, and grateful patients and families. And I would also like to thank members of my laboratory for their dedication. So, to motivate our study, we have to acknowledge that we have entered a new era of gene therapy for hearing loss. And just yesterday, FDA approved the first gene therapy for congenital hearing loss. And this news was featured by the New York Times last evening. And this gene therapy was approved for a very specific, rare cause of genetic hearing loss due to otoferlin mutation. So why is it that so many research groups across 4 continents have focused on this very rare type of genetic hearing loss? It's because the problem is limited to a very small portion of the cochlea, the synapse, and all other cells in the inner ear are intact. And this is a crucial differentiator because we know that gene therapy does not work if cells are not there. I'll just briefly mention that we have contributed to some of the developments that have led to the ongoing clinical trials, including characterization of ANC80, a synthetic adeno-associated viral vector, as efficiently transducing hair cells, not only in mice, but also in non-human primates, and developing the surgical approach that some companies are currently using that entails delivering gene therapy through the round window while making a fenestra in the oval window. But we know so little about the cellular basis of human hearing loss. And it's because, as you have heard from the prior panel, that knowledge comes from studying autopsy specimens, and that requires patients to donate their ears for study. And in fact, only 20 genetic causes of hearing loss have been characterized histopathologically, although we know that there are at least 200, and some argue up to 600 deafness-causing genes. So that's a big knowledge gap. Moreover, what we have learned from mouse models of human hearing loss, and these are mouse models that carry human deafness-causing mutations, is that in the vast majority of cases, degeneration tends to be progressive. So what starts as a defect in one cell type over time can affect many different cell types. This is illustrated in an example of a mouse that carries a human deafness-causing mutation encoding for connexin 26 protein. And as you can see in the left image, this mouse is born with a normal complement of cells in the inner ear. But by the time they reach adolescence, the organ of Corti is gone and spiral ganglion neurons are decimated. So it means that if you see a patient with this mutation when they are a newborn, a simple gene replacement therapy will work. But if you see them when they are a teenager, it won't, because the ear needs much more regeneration. So for all these reasons, we have really focused on enabling technologies that allow visualization of inner ear pathology on a cellular level, ultimately in living people. But why has this been such a difficult problem? This picture of the human temporal bone reminds us that the cochlea, shown here spiraling, is a small and delicate organ encased in the densest bone in the body. And in fact, today, what we know about the cellular origins of human hearing loss comes from studying autopsy specimens. When the temporal bone is extracted from the skull, it's decalcified, embedded, sectioned, and stained to generate sections like this. And here you can recognize that the human cochlea in cross-section is the size of Lincoln's upper face on a penny. So it's because of this small size and complex three-dimensional anatomy, encasement in the densest bone in the body, that today's clinical state-of-the-art imaging of the inner ear is very disappointing. You can see a computed tomography scan on the left that nicely outlines the bone, the OD capsule in white, or the MRI imaging that highlights the fluid-filled spaces in the inner ear, but we don't see individual cells. And that's why, over the last decade, we have focused on enabling optical coherence tomography to enable visualization of intracochlear microanatomy. So I'll very quickly summarize a decade of work in collaboration with my colleague Gary Tierney at Massachusetts General Hospital. Initially, we started with guinea pigs where we extracted the inner ear out of the temporal bone, made a little tiny cochleostomy, imaged through it, and could appreciate the basilar membrane and the vibrating organ of Corti, as well as the tunnels. So that emboldened us to do the next steps to see, can this technology be used to differentiate between pathology and normal anatomy? So for that, we exposed mice to loud noise, the equivalent of attending a loud rock concert for a couple of hours without wearing earplugs. And you can see a striking difference between the presence of the organ of Corti that includes multiple different cell types and its absence, which has dramatic consequences for future therapies, because we know that gene therapy and other therapies will not work in the scenario like this one, because there is the so-called flat epithelium and most cells are gone. So then the next step has been to enable microcochlear endoscopy by developing a catheter that can be inserted into the inner ear through the round window, the only non-bony opening into the inner ear, the same way we insert cochlear implants. And a few years ago, we published this pilot study showing that we can see the difference between the presence on the left and the absence of the organ of Corti using optical coherence tomography. And the histological equivalents are shown at the bottom. Again, this has dramatic implications for gene therapy and all emerging therapies out there. So the next question is, well, can we take it a step further? Can we see even more clearly? And more importantly, can we detect functional changes that precede structural changes? And the answer is yes. And this is where dynamic micro-optical coherence tomography comes handy, where dynamic really refers to the ability to detect intracellular vibrations of organelles that is ATP dependent. And it also allows us to visualize intracellular polymerization. So here, using dynamic micro-optical coherence tomography applied to the mouse organ of Corti, we can see individual cells. Look at these. These are individual hair cells. And if you need some help getting oriented to the anatomy, it's important to recognize that all of this is done without any contrast dyes. This is exclusively reflecting optical properties of the tissue. So we are now in the process of incorporating that technology into a microcatheter, and we're still analyzing the data, and I hope to show you some super exciting results in not too distant of a future. But in the meantime, we are looking at ways to validate this technology. And before that, here I'm showing you that we can indeed detect functional changes that precede structural changes, because after we expose this tissue to formalin, you can see that cell nuclei that were previously green have now turned blue. And that is really cool, because if we can detect functional changes, then it's opportunity to intervene when it's much easier to rescue cells rather than waiting for them to disappear or be nearly dead. So to validate our findings, we have been looking for technologies that would obviate the need for very time and labor intensive standard temporal bone processing, which requires decalcification, embedding, and sectioning. So a few years ago, we and others identified synchrotron radiation phase contrast imaging as promising because, as you can see on this picture of a human temporal bone, that's three-dimensional intact. We can see a beautifully spiraling organ of Corti, and we see rows of cells. Here you can see 3 rows of outer hair cells and a row of inner hair cells. And while this is really nice, it's very impractical because the current iteration of synchrotrons require a football-size synchrotron. So in the North American continent, there are only a couple of facilities that offer this for biological specimens, and understandably, they are overbooked, and it's very difficult to find time to image. So that's why we have been looking for more practical solutions and have identified X-ray microscopy. And here, this is a picture of a three-dimensional intact human temporal bone. And you can see the spiraling medaillus in the center. And now, as this video continues to unfold, you see these nerve fiber bundles that are beautifully rising up to innervate rows of hair cells that are continuing to spiral around, and you can also appreciate the lateral wall along with the stria vascularis. If we now take a view at the level of the basilar membrane, then we can see a virtual whole mount of the cochlea. Again, this is a human specimen, three-dimensional intact, and you see these beautiful rows of hair cells. You even see a tunnel of Corti shown here, as well as nerve fiber bundles that innervated. So in summary, what I've shown you today is that micro-optical coherence tomography is very promising, and indeed provides 100-fold higher resolution than the current MRI and CT scans used in clinical practice. And this will enable not only gene therapy for much more common causes of genetic hearing loss, but also other therapies that may involve cellular transplantation. And I'll just briefly mention that we have recently developed a protocol to make human spiral ganglion neurons out of tiny skin biopsies from a patient. And if we are envisioning transplanting these cells into live patients, it's important to know where to place them. So this is where illuminating the path will come handy. Thank you.